Hello, everybody, and welcome to another Expert Talk series coming right to your living room. Sometimes we give, and sometimes we ask for a gift. And that gift from each one of you would be to join our speech study. If you have ALS, if you have probable ALS, or if you don't have ALS, but you'd like to spend 15 minutes once a month for 5 months, it would make a huge difference in this journey for everybody's benefit. I'll turn it over to you, Christian. Welcome.
Thank you so much, and thanks everybody for joining. Indu has joined us as well. Thanks for everybody for coming tonight. And we're here this time around for our next expert talk in our expert talk series. And tonight we're joined by Dr. Ravindranath, where he'll be talking about targeting an endogenous retrovirus as a therapeutic approach to ALS. And just as a little bit of background, a couple things. One is just housekeeping. We will, of course, as always, take your questions throughout the program, and we'll address them in our Q&A section at the end of the presentation. Please use the chat to do so if need be. And if you have any requests for resources or anything, our team is here on hand to help out with that, as are your fellow Pathfinders and peers here who are always here to help as well. With that, I'm just going to give a little bit of an introduction to Dr. Nath, and the floor will be his. So Dr. Ravindranath is the clinical director of the National Institute of Neurological Disorders and Stroke, NINDS, at the NIH, where he is also chief of the section of infections of the nervous system and the director of the Translational Center for Neurological Sciences. He's a physician scientist whose research has been focused on understanding how viral infections lead to neurodegenerative diseases. While studying the effects of HIV on the brain, his laboratory discovered that a retrovirus embedded in the human genome gets activated in patients with ALS. And over the last 2 decades, his laboratory has studied the mechanisms by which this virus is regulated in the brain, the role it plays in pathophysiology of ALS, and is developing several therapeutic approaches to block the virus for treatment of ALS. His research in neurovirology has been recognized by the Pioneer Award from the International Society of Neurovirology and the Soriano Award from the American Neurological Association. And he's also received the Scientific Achievement Award from the World Federation of Neurology and the Presidential Award from the American Academy of Neurology. He's an elected member of the National Academy of Sciences, and in 2024, he was named by Time Magazine as amongst the 100 most influential individuals in health. It's a real privilege and an honor to have him join us tonight, and I know Dr. Nath is really excited to get to know the community as well. So with that, Dr. Nath, the floor is yours, and we'll hand you control of the presentation.
Well, thank you very much for the very kind introduction. I was really looking forward to this opportunity to talk to you. So I've put this talk together, but some of these things may be a little bit more mechanistic, so feel free to interject and ask questions. I think that would be perfectly fine, okay, to do so. So let's see. I won't be able to see the chat till the end, so you'll have to unmute yourself.
We will, the team will help you out with that as we receive questions, and we'll help moderate those with you.
Okay, thanks. Okay. Okay. Do you see my slides in presentation mode?
Yes, we do.
Oh, okay, great. Thanks. Okay, so for those of you who have not been to the NIH, we're just outside of Washington, D.C. And it's the intramural research program of the National Institutes of Health. Most people think NIH just gives out money to others to research, but actually it has its own research program on a campus here that was donated by a family. The entire land was really donated to the government, and they built this beautiful facility. These 3 buildings that you see here are is the hospital at the NIH. And so it's a research hospital. It's the largest research hospital in the world. And the interesting thing is that we can bring in patients, but they can only come on a research protocol. We don't have an emergency room, so nobody can really just walk in here. But we also have research labs that are connected to it. So my offices and my lab is in the hospital itself. So it gives the ease of seeing patients and bringing samples back and forth. And so you can do the kinds of research that is not possible in other institutions, at least easily. So I'm going to talk about these retroviruses. The term retrovirus, you may have heard from the AIDS pandemic. You know, HIV that causes AIDS is a retrovirus. And that's what most people think of when they talk about retroviruses. But it comes to surprise to most people to learn that if you look at the human genome, about 8% of the human genome is full of retroviral sequences. Okay. And what most people study are the proteins that are being formed by the human genome. Only 3% of the human genome forms proteins. Okay. And that's about it. So you have 3 times the protein coding region that forms retroviruses, and not a lot of people study them in the context of any kind of disease. Okay. So it remains a largely unexplored area, but I'll try to convince you that this is a very important area to study in the context of neurological diseases, particularly ALS. Now, how did they get in our genome? They've taken place throughout the process of evolution. So the last retrovirus that was integrated into the human genome is called HML2. And this is a virus that is a subfamily of a larger family of viruses called HERV-K. And the last insertion was around 150,000 years ago. So that one insertion of HML2 in the human genome is relatively intact, not completely intact, however. So the other interesting thing about these viruses are that just like HIV, all these retroviruses are sexually transmitted. And the question is, what is the advantage of these viruses to transmit sexually? And what they're trying to do is they're trying to get into the spermatozoa. Okay. Once they get into the spermatozoa, then the new fertilized egg will have this viral sequence that it never had before. And that's what leads to the process of evolution of species. So the kinds of retroviruses in humans are very different from the retroviruses in mice, for example. You know,
You said that.
okay.
Sorry.
So, we think that mouse models are important for studying human disease, but people who study retroviruses will tell you that the retroviruses in mice are very different than humans. And that's what makes a mouse and a human a human. Okay, it's not the protein coding region, which is about 90% about the same. The other interesting thing about, if you look at the process of evolution from primates to humans, is that the human brain is the largest compared in size and the most complex. Okay, so the other important thing of acquiring these retroviruses is that it also participates in normal brain development. And if it's important in normal brain development, then if something goes wrong, it can also cause disease, right? So the reason it's important in development is that it's expressed at very high levels in stem cells. So these round cells here are stem cells, and all this red staining here is the envelope protein of the virus itself. Okay. And so in early embryonic development, you need every single part of your gene because you're forming new organs, new cells. So you need everything that you can get, right? And once the organs have formed, a lot of these retroviruses will shut down because you don't need them any longer. And that's exactly what we found. When we took these pluripotent stem cells, we find here the envelope is expressed at very high levels. And then if you go neuronal progenitor cells or neurons, it kind of shuts down. Okay, so just as shown over here that you okay. So we also try to understand that what does it do at the stem cell stage, and I think important discovery that we made here is that it has a receptor, and this receptor is CD98 heavy chain. And these interactions between the envelope and its receptor here are critically important to maintain the stem cells in the stem cell stage. So it does that through a variety of secondary signaling mechanisms. Okay. But yeah, remember this CD98 is going to be important further when I talk about pathogenesis of ALS. So I told you that this virus shuts down in neurons. So the obvious question that anybody would like to ask is that, okay, what would happen if we force the expression of HERV-K in neurons, right? And so that's exactly what we did. We said that, you know what, we're going to make why don't we take a mouse, and what we will do is we'll force the expression of it in neurons in mice. So we took this promoter called Thi1 promoter, and that forces the expression in all neurons within the brain, okay, all neurons. And so what we did was, sure enough, you look at the transgenic mice by Western blot here, we can see the protein. And when you look at these mice, we found that their survival decreased, so that by 9 to 10 months, 50% of these mice were dead. Okay, so something's killing them. So we started looking at these mice further, and what we found was that the only abnormality in these mice were motor abnormalities. Okay. And so we said, okay, why don't we look at the lower motor neuron? There are many different ways of looking at the motor neuron, lower motor neuron. The lower motor neuron is the one that starts in the spinal cord and goes into the muscle. So if you look at these mice, they have this little hunched back over here. We looked at their muscle, and on the left-hand side, you can see this checkerboard pattern. You have two different type of muscle fibers, and they're taking a checkerboard pattern. Here, what you see is a neurogenic atrophy. So you lose that checkerboard pattern because there's some neurons are lost, few are left. The ones that are left, they're taking over the function of the dead ones. So you lose that checkerboard pattern. So that's a very characteristic muscle pathology of neurogenic degeneration. When we looked at the motor activity of these mice, you can see that they should cross the checkerboard, but actually what they do is they stay on the sides because they're impaired. They can't really run or walk very well. Then we said, okay, let's look at the brain. And so we looked at the brain over here, and we looked at the motor region. And what we found was that, yes, there's good expression of HERV-K in these pyramidal neurons. But when you looked at the neurons more closely, we found they're really abnormal. So a normal neuron should have all these wonderful processes here. And these ones, you can see all these punctae here. And there's loss of processes. So these neurons are highly abnormal. But the abnormality was only of the motor neurons. The other neurons in the brain were fine. And when you did testing on them, we found that the sensory vestibular system was all very good. When we looked at the spinal cord, you can see all these motor neurons here, and the wild type mice look just perfectly fine. But when we looked at the transgenic animals, you can see these anterior horn cells, which are the motor neurons. Most of them are gone. The few that are left here are really very, very large. And they're large because they're taking over the function of the dead ones. Okay. So the selective loss is actually very characteristic of ALS. Another hallmark of ALS is this protein called TDP43. Okay. And so it was discovered that this protein, the name TDP43 comes from TAT binding protein. So this was discovered in the context of HIV, which is a retrovirus, and it was very important in viral activation. But now people study it mainly in the context of ALS, because what they found was that if you look at the neurons, you will find them sitting in the cytoplasm. Normally, it should be in the nucleus. So if you look at this wild type mouse here, what you see is that all this green staining here is all within the nucleus. Okay. However, in the transgenic mice, it's in the cytoplasm. So that tells you that the expression of the envelope in these mice is forcing this protein to sit in the outside. So all the hallmarks of the pathology of ALS can be reproduced by overexpression of the envelope protein. So another question we asked was that if that is the case, can you actually detect the envelope protein in patients with ALS, right? So this is in collaboration with a company called Gen Neuro. So we send them some spinal fluid from our patients, and they did a blinded analysis and send us the data. When we looked at it, it was actually very, very interesting. So they do these chromatograms, okay? And then they take these chromatograms and produce these pseudo gels. So these are digitally produced gels. That's why they look very sharp, okay? So what they found is that this is the recombinant protein is the first lane, and that's the spinal fluid from two different individuals. And you can see that there's this envelope protein that you can find. And so they looked at a total of 14 different patients here from us, and you can see there's variable amounts of protein that is present. Some have more, some have less, One individual had none. And then you can, because it's a chromatogram, you can actually produce these numbers. So the level of detection is 10 nanograms per mil. And so two individuals was less than 10. The rest of them, you can actually find it. These were healthy controls that we send them. And one individual had a small amount of protein, but the rest we couldn't detect anything. Okay. So, okay, well, the spinal fluid has this protein. So if you take the spinal fluid and you put it on neurons, what would actually happen? Okay. So we put these neurons in culture, they're stained red, and you have this electrical grid there that then measures the electrical potential of these neurons, and it can measure the firing rate. So these neurons spontaneously fire. Okay. But if you just look at the morphology, what you see is that this is what it looks like with the media. Here we have three different spinal fluids. The spinal fluid from this patient number 9, it actually made a hole over here, it just killed the cells. Okay. And then you have variable amounts of toxicity here. And here's the firing rate, and you can see the firing rate for this is what you see in media, and then it decreases in all three of them, and number 9 is the least, right? And it causes the most toxicity. But what is interesting about this is that Gen Neuro gave us this antibody called K01, which is an antibody they made. They like to use it therapeutically, and what it is, that blocks the epitope on the envelope that causes neurotoxicity. Okay, so when you do that with the spinal fluid, you see that it protects against the toxicity of the spinal fluid, and it did that both for CSF3 and for CSF9. Right? Said, okay, well, you have the protein, and if the protein is there, can patients make antibodies to it? Can you actually detect antibodies to the envelope protein? And the answer is yes. So our hypothesis was that if you're producing a lot of the protein in these patients, you're going to have a lot of antibody. To our surprise, we actually found the total opposite. What we found was that if you look at individuals, here we categorize them by their survival. Okay, so individuals who survived for a very short period of time, individuals who survived for a very long period of time. Okay, and if you look at the titer of antibodies, what we find is that those individuals who had very fast progression of their disease actually had very low levels of antibody. Okay, and those who had long survival had higher titers of antibody. So that suggests to us that the antibody might actually be protective. Mm. Um, so now there are many different strategies that we and others are developing to try and block it. Okay, so a lot of this is all in preclinical studies at the moment. So we've put in a huge amount of effort trying to develop a gene therapy whereby we make these shRNAs, which is a way of blocking the RNA. It binds to the RNA of the envelope, and that way to prevent the production of the protein itself. Okay. Then Gen Neuro has made these envelope antibodies that I just showed you. So they have made a monoclonal antibody that you would have to give as passive immunization once a month or so. Then there are protease inhibitors. So there are protease inhibitors of HIV that one can already use. They're not really the best. So we're trying to develop, I have a medicinal chemistry group that is developing more targeted protease inhibitors that would interact and block the production of these viral proteins, okay, or at least the cleavage of the viral proteins or maturation. Then there are reverse transcriptase inhibitors. There are a number of HIV ones that are available, but there's a company called Rome Therapeutics. They have been working with Gilead in order to develop better reverse transcriptase inhibitors that are targeted against HERV-K. And then there are integrase inhibitors. And so we are just designing some experiments to start looking at newer integrase inhibitors. So that's relatively new. So these lessons we've learned from HIV because they're both retroviruses, and HIV, you've successfully been able to control it, and we know that combination therapy is a really good way to go. Okay, so when we published our very first paper showing that these retroviruses are important in ALS, there was a infectious disease expert, Julian Gold in Australia. He actually showed up in my office, and he says that, you know what, it would be totally unethical to treat patients not treat patients with antiretrovirals, and that we should treat all ALS patients with antiretrovirals. And I said, well, I'm glad you like our work, but I wouldn't suggest doing that because despite it being a terrible disease, and despite the fact that we have antiretrovirals, I know from experience that what can happen is that if you don't do it in the context of a clinical trial, you can end up hurting patients. So things that work in one disease can actually do the opposite in another, and we've seen that many times over. So he said, okay, we will do an open label study, which is exactly what he did. He took Trimec, and I showed him all the data on each of these drugs that we had in the context of ALS, curve K. And Beckauer had a really good profile. So he decided to treat them with Trimec, and they followed 40 patients. And what they showed was that's an open label study, so one's got to take it with a pinch of salt. And but what the ALS docs usually do is they will say that, you know what, we follow our patients for a while with this ALS score. 48 is a normal ALS score, okay? And anything below that means that there is impairment. So what they follow them for a while, and they found that, okay, these patients collectively are following this trajectory. If you draw a straight line, this is what you would expect them to go. Not everybody follows a straight line. Some patients have various kinds of variabilities. And what they said was, okay, those individuals who are on the antiretroviral therapy, they have a little bit of advantage over the others. Nobody was cured, nobody's disease was reversed, but they showed maybe there is some benefit here. Then they looked to see what domains are affected. They didn't see much in the bulbar domain. They found that maybe the motor domain makes some kind of a difference, and maybe in the respiratory domain. Okay. So now they're doing a double-blind study with Trimec. Okay, it's a much larger study, and they've enrolled all the patients, and the study is still ongoing. We decided to do our own study. Oh, yeah, let me show you the data for the Trimec then. So what we did was we asked them to send the sera to us. Okay, so we analyzed the sera of these patients, and what we found was we measured the HERV-K levels in the serum. And what we found was that over 24 weeks, there is a slight decrease in the levels. So the levels never came down to zero. There was a decrease in the level. And then what happened was when they went off the drug, you saw a rebound. This is within 1 to 2 weeks. So very rapid rebound. So that's what really gave me confidence that these drugs are probably doing something. Yeah. So what we said was, okay, now not everybody is levels are going down. Why don't we look and see whose levels went down and whose did not, and is there a difference between the two? So we said, okay, the non-viral responders are those individuals the level didn't change or went up. Responders in which the levels actually went down. So if you look at the responders in which the levels went down and you look at their ALS score, you find that most of these individuals, number one, they were doing relatively well to begin with. They didn't change a whole lot. The individual who wasn't doing that well actually went down pretty fast. If you look at the ones in which the levels really did not change, you do have about 2, 3 individuals didn't change very much, but most of the others continued to progress. Again, it's an open label study, small sample size, so you got to be careful in how you interpret it. So we decided to do our own antiretroviral study, and this study was approved and started on April 1st, so as close to March as you can get. So we called it the NIH Combined Antiretroviral Study, or the NCAA study. Right? Okay, so here we decided to use a different combination of drugs, and we had a different inclusion criteria. They took all comers. We decided we were going to pre-screen individuals, and only those individuals who had high levels of HERV-K were we going to take. So our cutoff point was 13. So unless you have 13 or above, you weren't going to enter the study. And we decided to treat them with 4 drugs. So we used TAF, which is a reverse transcriptase inhibitor. We think this is actually a much better reverse transcriptase inhibitor because it accumulates in the brain. We used dolutegravir, which is also in Trimec. We used durinavir, which is a protease inhibitor. And the protease inhibitors are important because they prevent the GAG, and the pol. The pol gene forms the reverse transcriptase for that protein to cleave. Unless that protein cleaves, you're never going to have any reverse transcriptase activity. So we thought this is going to be very important. It's an extremely potent drug. So we actually use this combination. Ritonavir is always used in combination with darunavir because you need to boost its levels. It blocks the enzyme in the liver, which would otherwise degrade the darunavir. Okay. Okay, so now this is our data. This remains unpublished. So here what we did was we looked at the HERV-K levels. As you can see, ours are very tightly controlled because we didn't take anybody who had less than 13. So, they cluster really nicely. And you can see that there was a significant drop even at 12 weeks. And it gets even further down at 24 weeks. Okay, so there's a progressive decrease in level, and it's statistically significant at both time points. When we took the patients off the drug, there's a rebound, but it's not that big a rebound as we saw in the Trimix study, and this is 4 weeks later. And I think that's probably because the type of drugs that we chose, they have a much longer lasting effect. Okay. Now, the other interesting thing we did was instead of looking at the here we're looking at proviral DNA. So we also collaborated with another company called Neurodex. And what they did was they looked at exosomes. So these are things, particles you can find in the blood or serum that are derived from the brain. Okay, so these are neuron-derived exosomes. So that's about as closest as you can get to measure it in the brain of an individual, but yet the fluid is coming out of the blood. Okay. And so when they looked at here, they measured the HERV-K RNA in serum, and they found the exact same thing. You can see that it goes down at 24 weeks, comes up at 28. And the other interesting thing they could measure was something called TDP43. I showed you earlier that's a critical protein. And what they found was that goes down too over 24 and then 28. So the interesting thing is that never rebounded. It continued to actually go down. So I think the half-life of this thing must be very, very different. Yeah. Okay. So we tried to also do the same thing as we've done with the Trimix study to look at responders and non-responders. We first of all put them all together in one single graph. And it's really hard to really make a lot out of it. So visually, you can see that people follow different courses. Some don't progress that much, some progress really fast. Some total, this is before entry into the study, and then after that, maybe there's very slight stabilization. I don't know. But I won't put too much weight on it. Then here, because we selected our patients so well, our non-responders were only 5. So our sample size is so small with the non-responders that it's hard to interpret. But nonetheless, we found that there's some responders. You can see most of them did well. It's pretty much like we saw in the Trimix study. And those individuals had really poor scores. They also didn't do all that well. Right? You can see over here too. They have bad scores. They did poorly. So, as I told you, one of the things we're very keenly interested in is to really develop a gene therapy. And I think that's much better than the drugs. The drugs are not optimal. And I showed you in 2 different studies that do something, but they're not stopping the disease. Okay. With the gene therapy, where you have the ability to block it completely. Okay. So there's absolutely no expression of the protein any longer. Okay. So what we did was we put it in this adeno-associated viral vector. And this is just to show you that, yes, we have really good expression of the viral vector itself, and we are able to block the expression of the protein and by using each of them individually, the 3 of them that we put in there in tandem together. So that's the other interesting thing about our approach is that there's not just one RNA, but this shRNA, we actually have 3 of them together, okay, in one single construct. So we can block all the different epitopes because there are multiple copies of the virus in our human genome, and we want to make sure we block them all. Okay, so we give it to our transgenic mice, and we say, okay, does it do anything? And so one of the things you can do with these mice is you can put them on this rotor rod. Mice love to run, okay? So you put them on a little wheel, and they'll run. And then what you do is you keep increasing the speed of this rotor rod and see when the mouse will fall. So and what we found was that so you have the wild type and you have the transgenic, and then you have the ones that have the shRNA or microRNA against the envelope here in the red. So if you compare the blue to the red over here, okay, this is the one that did not have that in the gene therapy, and the red one did. you can see that the red one actually did a whole lot better than the blue one, right? And then and we can also lift the mouse by its tail and see what happens to the hind limbs. This is a measure of spasticity. So it's an upper motor neuron side. So what happens is that if the mouse has spasticity, the angle between the two limbs will be very short, okay? Because like they're collapsing together. And if they splay out, that means that there's loss of tone, okay? So you can see that the envelope mice, the angle was actually much shorter, showing that there was in the transgenic mice, showing that there was spasticity, and the ones that were treated, they're coming back close to normal. Okay. And then we also looked at the muscle, and what we found with the muscle was the same thing. So if you look at the wild type mice, you see this really nice checkerboard pattern over here. And um, um, and then in the transgenic mice, in the control mice, you lose this checkerboard pattern. Yeah, you don't see that over here. And when you treat these mice with the gene therapy, you can see that it returns again. Okay, so in the upper panel. So we looked at two different types of muscle fibers, and you see the exact same thing. So it prevented the degeneration of both upper and lower motor neurons. All the injection was made direct to the brain of the mice, I mean, to the ventricles of the mice.
Okay.
Okay. And we looked at the spinal cord, and we found the same thing here. So we found that number one, there was really nice delivery of the AAV9. You can find good delivery in the spinal cord. So and that's very important because we need to know whether this virus the AAV9 will distribute amongst the brain and the spinal cord or not, right? And we found that it blocks the production of the envelope also. Okay, so we also found that it blocks the protein. We measured the amount of protein there, and also found that the protein is also neurons that are expressing the protein also get protected by doing so. There's loss. Sorry here. Yeah, this is the control. This is the transgenic with the envelope here, and yeah. So percentage of envelope neurons. Yeah, that's somewhat decreased here. Yeah, I forget. Okay. Oh, yeah. So that's exactly what you expect. So, and I understand. So what we were doing here is that in the transgenic mice, you have a lot of expression of the protein. And if you give the gene therapy, what you expect to see is that the protein should go down, right? And so that's exactly what we found. So the amount of protein has actually gone down because the gene therapy is blocking the production of this protein. Okay, so these are the people who actually do the work. And so we have a large group of people who are working on different aspects of this thing here. So I have a group that is working on just understanding the biology of this virus in the context of neurodevelopment. This is a group that really is very good in studying neurotoxicity. So all the data was really produced by them. And then Minghua did all the animal work. And then there's huge amount of work that we've done. I didn't show you. It's just understanding the genetics of these viruses in the human genome as to where they're located, how they get expressed, which loci are being expressed in ALS, and the like. And then this is a group that screened a lot of antiretroviral drugs in vitro. And then these are other industry collaborators that are working on various aspects of it. And Tony James is working on neuroinflammation, trying to see what happens to inflammatory changes in the brain in the context of it. And then how the expression is actually regulated is Jack Marshall is doing this stuff. And the immune response to it was all studied by Marta. And this is our clinical team that's been doing the clinical trial. So I'll stop here and take any questions that you might have.
All right. Thank you so much, Dr. Nath. We do have a couple of questions. So first question, since you have originally found that higher HML2 ENV antibody levels correlated with longer survival, do HERV-K, HERV-K antibody levels change differently in responders versus non-responders?
I haven't measured that yet. That would be a good thing to do.
Okay. Thank you.
So, hello, Dr. Nath. This is excellent presentation, and thank you for addressing a lot of the questions that people have. And we do have Dr. Ostro on the call here, who's our key scientific advisor. So, Lyle, you can ask your question here.
Actually, Christian just did. That was my question. Indeed.
Okay.
I'm curious about whether the antibody levels identify responders or non-responders since they correlated with, but I think, Avi, you just said you haven't looked at that yet, right?
Yeah. No, hi, Lyle. Thanks very much. That's an important question. We have the ability to do it, but we just haven't been able.
And then the other comment I made is just that it's always just really inspiring to see how this has developed from the basic science to these trials. And in particular, when we keep hearing in this landscape about trials that aren't meeting their endpoints, that the effort that you've put into identifying predictive biomarkers to figure out who should get this and how you can tell if it's working, since we know that, there's many different flavors of ALS. So I think I think personally, one of the things I find most exciting about this work is that you're figuring out who to give it to and how to tell if it's having the biological effect it's supposed to have.
I mean, thanks very much for bringing that up. So, that's what I've spent a lot of my career trying to understand why clinical trials fail. And a lot of pharmaceutical companies do these very large trials, and most often they don't show any benefit. So they're very disappointing. And the reason, at least in my mind, is that they don't stratify their patient population based on what they are trying to really target. And with neurological diseases, these are clinical diagnoses and they're heterogeneous diseases. So whether it's Alzheimer's or Parkinson's or whatever you want to study, there's not a single disease. They're all heterogeneous. So you really have to define your population. And if you can define them well, you can actually show efficacy in a smaller sample size. You don't need hundreds and hundreds of patients to do a clinical trial. So we've put a lot of effort into thinking about how to stratify patients. And so thanks very much for bringing that up.
Perfect. So the next question is, how would this kind of retrovirus therapy be administered with people with ALS? And is there an upcoming trial?
Yeah. So, I mean, if you just want to go with antiretroviral drugs that already exist, though they'll be all oral, right? If you're going to go with one of the gene therapies, I showed you the adeno-associated viral vector. We've also got antisense oligonucleotides. Those have to be delivered parenterally. So that means we will be giving them through a lumbar puncture. So we'll do that, or what we call as a cisternal puncture. That's another way that you can do it. So that way you can get better distribution. So we might even consider giving 2 injections, one in the lumbar region and one right at the base of the neck. So yeah, those would be now if you go with the antibody, so the antibody is an intravenous injection. So those will be monthly intravenous injections. The gene therapy will be a single injection if you go with the AAV route. If you go with the antisense oligonucleotides, you will need repeated injections.
Thank you.
Thank you. Next question, Dr. Nath. Are there any adjuvant drugs that enhance the effect of antiviral therapy that you're testing?
Does it like to enhance the effect of the antiretrovirals? So
Really
yes,
any-
it is possible you could enhance it. So a lot of these antiretrovirals are PGP substrates, which means that their uptake into the brain is not the greatest. The brain endothelium will actively push it out. So there is a possibility of using drugs that block the PGP molecule, and that way the drug cannot be pumped out of the brain and actually stay there.
Thank you. Next question. Can any antiretroviral therapy be used in the meanwhile, since we believe we are far away from this therapy reaching patients that may have limited time?
So I think that's fair. That's a fair question to ask. We did these 2 open label studies to make sure that these drugs were going to be safe and that we don't end up hurting people. So I think, and these drugs have a very large safety record. There have been a lot of patients with HIV infection have been exposed to it. number of drugs are actually given to healthy individuals now for prevention of HIV, so they can be used prophylactically. So I think they are safe to use. I think is it going to do anything for anybody? That would be harder to predict because what we do not have is a commercial assay to really measure the viral load. So we have a research assay, but we don't have the ability to, and our IRB won't even allow us to just measure it in the context of just treating patients for clinical care purposes. But I think if somebody were to commercialize the assay, it would make it a lot easier to monitor patients. But yes, there's no harm in using these drugs. They're relatively safe. The side effects really are GI side effects. That's actually something to remember. And that's was our problem here is nausea and for some it's loose stools. So the nausea was transient in our study, and we did put a number of patients on anti-nausea drugs. So I think that would be an important thing to, if somebody is deciding to go on these drugs, to make sure you manage that nausea properly. And do it in the context of some physician who's closely monitoring you, and knows these drugs.
Thank you. Thank you, Dr. Nath. So the next question is, with a lot of AI drug development buzz to enhance or to accelerate some of these findings, are you using AI to enhance, likelihood of success or better target for subtyping?
Yeah. So I'm no expert at it. I am very curious myself as to how we can use those tools for drug development, but sorry, I don't have any expertise in it. So we haven't gone that route yet.
Dr. Nath, next question is, thank you for that. The next question is, do most ALS patients have or HERV expression?
No. So at autopsy, we found about 50% had expression. And when we tested the patients, we think about 30% of them have HERV-K expression. What we did find, however, is because some of our patients who didn't meet inclusion criteria, we continued to follow them to see what happens to their HERV-K levels. And what we found was they go up over a period of time. So it's quite possible that, there are two possibilities here. One is that, yes, there may be more than 30% that express HERV-K level. Second is we may have missed the opportunity to treat people early in the disease, and we waited too long till the levels were high. Yeah.
Okay. Thank you.
So this question is regarding. Go ahead, Christian.
Okay. The next question is about Japanese pathogenicity and the studies of TDP-43 injections in primates cause disease. Would these TDP-43 preparations continue the virus?
I don't know what TDP-43 preparation they're talking about. They inject TDP-43 itself?
Yes. Yeah.
Well, I don't think that's a good idea. Some other ALS expert can probably mention. TDP-43 is a culprit. It's not a treatment. I don't know the rationale for putting TDP-43.
It's just going to TDP-43 in primates were actually causing disease.
Yeah, it'll cause disease. Yeah, that's what I would think. You don't want to treat people with it.
Thank you.
So do you have any thoughts on the data presented in Montreal at the MND conference that said that there is no difference in incident in Africans taking antiviral for HIV?
Yeah, I don't know that data. Yeah. I have no idea. And I don't know how you would actually figure that out. You need a large database. You need to figure out that the diagnosis was made properly. You need to know what drugs they were on, how long taking it for. You don't measure their HERV-K levels, so you don't really know. So I don't know. To me, that's
to clarify, I think that question was about that there's not a higher rate of ALS in people with HIV, right? Isn't that the data presented?
Yeah.
And yet there is. I mean, Dr. Nath, I think you're the one who taught me that there is something that looks a whole lot like ALS that can sometimes happen in people with HIV/AIDS and that goes away when you treat the HIV, right?
Yeah, so this is another fascinating thing. Thanks for bringing that up, Lal.
Yeah.
So that there are a subgroup of HIV patients who develop an ALS-like syndrome. And if you didn't know they had HIV, They look exactly like ALS. They are much younger because HIV occurs at a younger age group. And if you catch them early on, within the first 6 months or so, and you put them on antiretroviral drugs, you can actually reverse the disease. Tell you the neurons were not dead, they were just dysfunctional. If you treat them beyond a year of their symptoms, you can slow down the progression of the disease, but the disease doesn't halt or it doesn't stop. Okay. So I think early treatment is going to be important. Yeah, it's very true.
So I think the context of that other question was that there was some data presented, just epidemiologic data that said that if you look at Africans who have HIV who either are or are not taking antiretrovirals, that there was no difference in their ALS incidence. I'm not really sure how you would interpret that. So
Yeah, just as I said, it's meaningless
Yeah.
because you don't know what antiretrovirals they were taking. Not all antiretrovirals block HERV-K. You don't know whether the HERV-K levels were elevated or not. You don't know how long they were taking it for, what the compliance is. There's so many variables that you would need to know to make that kind of conclusion.
Thank you. There was another question also, I think, Dr. Ostro, you may have posed it, or somebody else in the audience did that. You've mentioned previously that the degree of inhibition necessary for treating ALS is greater than would be needed for HIV, and that this presumably would take adjusting the dose depending on viral load. Is this a reason you're favoring the other treatment modalities like gene therapies?
Yeah, so that's true. The thing is that the dosage that we're using is the exact same that is FDA approved for HIV patients, right? So we did not try to exceed that dosage. And at that dosage, the viral load comes down very gradually. If you give an HIV patient, you'll see the viral load will come down in a matter of few days. Boom, down. The ALS patients is taking 6 months for the level to come down. It's coming down very gradually, right? And even when it comes down, it doesn't bottom out completely. With HIV, you become undetectable if you take these antiretroviral drugs, right? Here, you never become undetectable. You can always still detect it. So they do something, but they're not the best. So that's why I think the gene therapy is going to be absolutely critical. Yeah.
Thank you. And another question about, well, trials in general. When will the next trial in humans be? Can we get these drugs now with a prescription from our physician, since the drugs are already available for other diseases and be off label, or do we need to wait for a formal trial?
Okay, so that's a tough question to answer. The drugs are available. Anybody can prescribe them. The thing is, I don't know. I mean, it's not an unethical thing to do. It's just the thing is that whether it really does anything for you or not is very hard for you to know. So we are interested in doing another study, a double-blind placebo-controlled study. And that study is still in the works of still writing the protocol up. So we don't even have IRB approval quite yet. There is a double-blind study being done by the Europeans and the Australians using Trimec, and they had 250 patients, I think, in each arm, and that's been fully enrolled. Okay, so those results should become available in a couple of years. They are treating them for a full year with antiretrovirals, which is a good idea. Now, their endpoint, unfortunately, is survival, and they didn't stratify their population based on HERV-K levels. They took all comers. So they enrolled very fast, but the downside of their study is that I don't think it's going to show any great effect because they'll have to do subgroup analyses to really figure it out if there's an effect or not. Because they've asked us to measure the levels, which we will do. And then I think you'll have to reanalyze their data to try and see the individuals who had levels and whose levels changed, did it really make a difference or not. So I think ultimately those answers will become available, but that's where we stand. And our study hasn't yet been published, so that's another thing. So that data is not widely available, although it was presented in Montreal. Yeah, in that sense, some of the data is out there.
Perfect.
Go ahead, India.
So there was another question on multiple myeloma. Have you seen any correlation between ALS and multiple myeloma and in HERV-K in both of the diseases?
Oh, that's a good question. So I think what this person is getting to is that their HERV-K has been associated with cancers, okay? Number of different types of cancers. And so I told you that it is expressed in stem cells, okay? And what it is, and it's very important in cell proliferation and organ formation, right? So if you were to now re-express it in a cell that can undergo mitosis, that means mitosis means cell division, you can actually cause cancer. Mhm. So if you were to take it in a prostate cell or a breast cell or something like that, you can actually push it to mitosis. It results in cancer. In a neuron, when you try to express it the same way, you can never push a neuron to mitosis. It actually dies. So you get neurodegeneration. That's what happens in ALS. But in these cancer patients, it is, if you look at the stem cells within the tumor itself, that's where you find the HERV-K expression. Now, I would be very curious to study individuals who have ALS and develop cancer at the same time. I think so. I've spread the word around, and what most people tell me is it's going to be very hard to find them because the life survival of ALS patients is so short, and the tumor, some of these tumors is also short, although longer than ALS, is that the two of them simultaneously would be very hard to occur because you may die from ALS a lot quicker before the tumor can actually form or to the point whereby it can be recognized, right? So, and expression in neurons may not equate expression in some other organ system. But if we could find individuals who have ALS plus some other disease, we would be very curious to study them.
So as a follow-up question then, Dr. Nath, is that how best can people with ALS test if they have HERV-K
We cannot
virus?
test. We cannot test it because there is no test available. So some company has to start doing it and make it commercially available. I think this company Neurodex is interested in doing it, but they haven't quite gotten to that stage yet. So it has to be made commercially available.
And tested in your lab, correct?
We cannot do, RB will not allow us to do that. We can only do it in the context of a research study,
Okay.
and we cannot do it for clinical care. So our IRB told me very, very sternly that I cannot do it.
There are lots of research tests to test for it, right? But that's the difference, is that testing for something in a research lab is not a certified test that can be done on people living with the disease in a certified way.
Thank you. I'm gonna say thank you.
Thank you. And so for next question, I guess relative to that is, do HERV-K positive ALS patients, as we were talking about its relationship with AIDS, HIV/AIDS, do they have more cognitive involvement, such as in AIDS dementia?
So sometimes it's hard to figure that out because HIV patients may have cognitive impairment just from the infection itself. But the ones that I have seen, and there are only a handful of patients, maybe about 10 or 12 is all that I've seen with HIV and ALS. I don't think they had much cognitive impairment. They was largely motor type ALS. They did not resemble FTD or anything like that. At least the HIV ones did not.
I think this person's maybe asking a different question, which is in the third of patients who have high HERV levels that are ALS patients, not HIV patients.
Oh.
Do the people who have high or low, for example, HERV levels, is there a more frequent cognitive involvement, like, FTD ALS, for example, as opposed to straight ALS? And I don't think there's any data for that, but I'm not.
Yeah, we'll need a larger sample size for that.
Yeah.
There is one paper by the Australian group claiming that they found HERV-K activation in frontotemporal dementia. Yeah. But yeah, we haven't looked at it.
And other high inflammatory components or diseases, especially somebody has asked about their type 1 diabetes and ALS. Is that of interest when you study HERV-K?
I don't see how the two would be related. Diabetes is such a common disease, although type 1 is not all that common. There you're looking at an autoimmune disease. So there is another retrovirus called HERV-W that's been associated with diabetes, but HERV-K is not, as far as I know.
Thank you. And we had excellent questions, and you inaugurated our 2025 Expert Talk series. So we're really excited with having these discussions moving forward, and good luck with everything you're doing. And we are citizen-driven research, and we love to work with you directly. If there is anything we can do, please let us know.
Certainly will. Thanks very much. It was really a pleasure to have the opportunity to talk to you. All great questions, and appreciate your interest in our work.
Thank you.
Thank you.
Okay.
Have a good evening.
Have a good evening.
Well, folks, another Right to Your Living Room. Great discussion. We all care. We all want the same success. ALS, out the door. But until then, we have each other. And to me, each of you are very special people. So thank you for sharing your life with us, because we definitely have our lives to share with you. And with that, let's see what kind of hearts we might come up with tonight as we say good night to each other. All righty, on 3, let's do it. 1, 2,
Good to see y'all, guys.
2025.
Night, Faith.
Hey, Joe. Good luck on that Alaska cruise.
Good night.
Good night.
Wow, appreciate
Thanks,
it.
Ellen.
Good night, Sean. Good night, Joe. Good night,
Wayne, we'll talk.
Indu. Good
Happy
night.
2025, everybody.
Susan, Ruben.
Happy
Love you, Susan. Okay. And
you,
with
Faith.
that, it's
Thank you,
really
Kathleen.
hard to ever press this button to say goodbye.